Good morning, everyone, and thank you for joining us in our expansion user meeting today. We are very lucky to have Dr. Ajay Labade joining us. He's from the Harvard Department of Stem Cell and Generation Biology from Jason Buenrostro lab. And today we are very lucky he'll be talking to us about his work in expansion and in situ genome sequencing. So I leave you the floor. Thank you, Ajay.
Thank you, Gabriela, for the invitation and nice introduction. I'm really grateful to talk about our research in front of this wonderful group. I looked into the website and it's really nice group about expansion microscopy. I also went to the Garvan Institute website, and it's really nice to see like people working in this field. Today, I'm going to talk about a technology called expansion in situ genome sequencing. So as you can see from the title, there are two important sections of this technology. One is expansion, which refers to expansion microscopy, and the second one is called in situ genome sequencing. I'm going to take you through this technology, the way the journey in which we develop this technology. And I'm gonna also tell you how we applied this technology to understand relatively unknown biology of a disease which is called as fast aging disease, progeria. And hopefully at the end of my talk, I'm gonna leave you with a really new perspective of looking towards genome at extremely high resolution, and also to look at the DNA sequences interacting with proteins inside the nucleus. But to begin with, I wanna ask why in situ genome sequencing? Why do we want to sequence genome inside the nucleus, inside an intact cell at extremely high resolution? And to start with, I have this beautiful figure from a paper called Chrome EMT, which was published in 2017, which allowed us to look at DNA and how it is packed inside the nucleus at extremely high resolution. And as the title suggests, it's the nanoscale architecture of our genome, where structure dictates function. So I want to stress on structure and how it dictates function. Evolution from the history, you would see that biologists have always used structures to classify cells, not just cells, but smaller organelles inside the cell by just looking at the structure, shape, and size. So that means structure somehow dictates function. And evolutionary, our cells have learned to read the structure, the information that is stored inside the structures. And the same is true with our genome. So the genome starts with DNA. DNA itself is a structure. Every base of the DNA is a structure. And somehow enzymes have learned to read that information which is stored in the DNA. But today, I also want to focus on the scale at which these different structures are formed. As we know, that the DNA itself is in the range of a couple of nanometers, if you look at the width of the DNA. And then it arranges in the form of nucleosomes by interacting with these histone proteins, which are positively charged proteins. The nucleosome forms a structure called chromatin, which is in the range of tens of nanometers. And if you look at an antibody that we use many times in our labs, it falls into this range of a few tens of nanometers. So one nucleosome can be bound by one antibody molecule. And then the chromatin forms higher order structures, as we know. Those are called as chromatin domains, smaller domains, nanoscale domains, and then larger domains. And then finally, they form chromosome territories. So what this paper allowed us to look at is how all of these different forms of chromatin are organized inside the genome. And they found that the DNA, there's it ranges from really like a small scale, which is few nanometers, to hundreds of nanometers. So traditionally, we have used different techniques to look at these structures and how different proteins are organized along with the DNA inside the nucleus. And one of them, as we all know, is microscopy. We use microscopy to look at the DNA. There are techniques called as chromosome paints, where you can stain the entire chromosome using certain small DNA probes. They can specifically go and bind to certain chromosome, and we can look at the entire chromosome. But that is in the range of hundreds of nanometers because these chromosome territories form large domains. Similarly, we can use fish probes, which are relatively small, but still we cannot visualize DNA, which is at a very, very small scale, which is in nanometers. So for the smaller scale, we have always used biochemical techniques. That includes chromatin immunoprecipitation or chromatin conformation capture methods, where we can capture the conformation of genome, how it's organized. But what it lacks is the spatial information, where that piece of DNA is coming from, or how that particular part of DNA is organized inside the nucleus. And therefore, we need a technique that can allow us to do both. The technique that can allow us to not just look at the DNA, but also to sequence the DNA. And therefore, we primarily had two questions to begin with when we started this project. First is, how can we resolve the genome at nanoscopic resolution? Because we need something that can allow us to look at extremely small structures, just like DNA or maybe a very small chromatic domains. And second, how can we create a multi-dimensional map, multimodal map, where we can not just look at the DNA, but we have to also look at proteins organized, interacting with that particular part of DNA at extremely small scale. Therefore, we decided to combine these two very interesting technologies that were developed by our lab. First is in situ genome sequencing. I'm going to talk about both of these together in my talk later on, but this in situ genome sequencing is a technique that allows to sequence the DNA right inside the cell using microscope. And expansion microscopy, as most of you are familiar with, is a technique that allows us to look at nanoscale structure inside the cell. So our challenge was to combine these two technologies in our method. And that is where we developed a technology called expansion in situ genome sequencing. I'm going to now first start with first problem. That is, how can we resolve the genome? Because previously expansion, before we started this project, expansion microscopy have always been used to look at large protein structures or cells, but not DNA or a genome per se. So our challenge was to resolve the genome, to expand the genome. So we started working on genome expansion, and we followed a very simple protocol that is always followed in expansion microscopy, but we characterized by many ways to show that the genome can be isotropically expanded without changing the spatial organization. So we fix cells and stain them with different chromatin antibodies. In this case, I have used speckles to label in green and then lamine in red. As you all know, in expansion microscopy, we embed these fixed and stained cells in an acrylamide gel. But before that, we treat cells with a chemical moiety which interacts with amine groups of proteins. So all the proteins are linked with this molecule called anchor molecule. In our case, we use MANHS, methyl acrylate NHS ester. So this adds the acrylate moiety, methyl acrylate, to every protein, every almost all lysine groups of proteins. And after the gel polymerization is done, what happens during gel polymerization is that all the proteins become part of the gel. So they are all polymerized along with the gel. So they are nothing but a gel polymer. Then we digest these proteins because proteins are structures, they have their own shape. We need to make sure that when we expand this gel, we have to digest the protein so that they won't anisotropically pull the gel in all directions. Therefore, we have to chop the protein into smaller peptides, and that is what we do in digestion using enzyme called proteinase K. And one of the good, like the best property of this gel, as we all know, is that when we put this gel in plain water, what happens is this gel is made up of sodium acrylate. The sodium ion repel each other when as soon as they are put into the gel is put into the water, and because of this repulsive force, it expands isotropically along with the protein moieties. So in this case, the protein is also antibody, which also has fluorophore on it. And since the protein was chopped, the antibody was chopped into smaller peptide, it isotropically expands, and we get a very, very good resolution of what is present inside the nucleus or a cell. So this is the expansion microscopy, but our challenge was to characterize whether it is causing any anisotropic change. So what we start with is unexpanded cells. So we take a large field of view of all the cells that we have on, let's say, for example, cover slip. We take a huge montage, and then we expand all those cells together, and computationally, we can then find out before and after expansion cells. And here I was able to poke this particular cell here with a needle. Uh, this image is just to show that there is a very nice isotropic expansion, and I have one more image to show you. So this image was taken at 40x objective before expansion in the pre-expansion section, and the same cell was then expanded in post-expansion section, and we can compare the resolution pre and post expansion, calculate the exact expansion factor. We can also look at nano changes in the genome. Let's say in this case, the DAPI stain, if there is any change, anisotropic change, and we didn't see much of a change. So we can expand the genome, DNA, but we wanted to validate more. So what we did was we used nuclear pores as a molecular rulers, because nuclear pore structure is very well known. Its dimension is well known by electron microscopy. So we wanted to compare our images with the electron microscopy images and see if we can isotropically expand these nuclear pores. And what we can see is that you can see here in green is a lamine and red are pores. And you can see right below the nuclear pore complex, we always see this heterochromatin exclusion zone, which has been shown by electron microscopy. In this image, you can see it very clearly that the nuclear pores are stained with red. And you can see here, it forms the outer ring and inner ring. As we know, the nuclear pore has both outer and inner ring. And we can resolve these two rings. That means we can get a very high resolution in terms of few nanometers. Not only that, but in gray color here, what you're seeing is a chromatin, which is DAPI. And you do see that right below the nuclear pore complex, there is a DAPI less region, which is this dark region, which depicts the nuclear pore. We can count total number of pores, and they resemble very well with the electron microscopy images or the counts. There are typically 2 to 3,000 pores on every normal cell. So this tells us that we are able to expand the chromatin isotropically. There is no change in the way it is organized at a very small scale, even at a nanoscale like a nuclear pore. Now comes the part of sequencing the genome. So we can now expand the chromatin, but how do we sequence it? There we use our technique, which is in situ genome sequencing. But the challenge was to combine it with the expansion microscopy. So what we start with in situ genome sequencing is we take this enzyme called TN5, which is transposase, a very active transposase. And one key characteristic of this enzyme is it is a doublet, it is a dimer, and we can load this dimer with a known DNA sequences. We call them adapters. And these known DNA sequences are then inserted into the genome by this enzyme. So it forms this dimer, goes into the nucleus, and wherever the DNA is free from proteins, it tagments it. Tagments means it cuts the DNA and insert these known adapters. Now, what we can do is we can put a known DNA sequence that we want to insert into the genome. So what we do is we take two different DNA sequences, AD1 and AD2, so that we know the sequence of these two barcodes, and these barcodes are then inserted into the genome by TN5. One trick that we use here is to open the DNA, because TN5 has inherent capability to only tagment open DNA and not the compact DNA. So what we do is we do HCl treatment that is regularly used in fish technologies, to open the genome. And once the HCl treatment is done, most of the genome is highly open, so that the TN5 is now can go and tagment the genome non-specifically everywhere. We do multiple rounds of tagmentation so that we get maximum number of DNA fragments tagmented. And remember, the cells are fixed, so the proteins are still there, fixed by formaldehyde. They form covalent bonds with the DNA, so the DNA still stays inside the nucleus, right? Even after the tagmentation. A very free fragment that we have noticed sometimes do move out, they diffuse out, but we do not care that less than 1% of the genome is in fact completely free of proteins. So after that tagmentation is done, as you can see, it has these adapters at the end, this green DNA label here. The next step that we do is, since we know the sequences that are attached, these green sequences attached to the genomic DNA, we can now insert dumbbells. So these are the dumbbell DNA sequences. They form this dumbbell shape. These are called as hairpins. And these hairpins have this complementary region that can go and hybridize with one of the adapter sequence. So we can now have these DNA dumbbells in the genome. So there is genomic DNA inside, and these hairpins are now ligated to both ends of the DNA. So you get genomic DNA and these dumbbells. So one key thing about these hairpins is that we can now insert a barcode. So this is a unique molecular identifier, a unknown DNA sequence, which you can order from IDT. So this has N, which are all mixed bases. So that what happens is every DNA fragment, genomic DNA fragment, is now barcoded with this barcode. And we do not know the barcode because every fragment is unique now. It has its own barcode. Other side of the dumbbell has a rolling circle amplification primer binding site, so that when we put the primer, we can perform rolling circle amplification on this dumbbell sequence, because you can see it is basically a circle of the DNA. But one thing that we did is that we wanted to further attach these DNA sequences to the gel, because now the next step that we do is form a gel. But before we form the gel, we hybridize a anchor sequence. This is called as hook, oligo hook. So it goes through this one particular hairpin, and what it has on both sides is a acridite moiety. So you can get these acridited oligos from IDT. So now what happens is when you form a gel on this tagmented genome, it is going to get linked to the gel, so that when you expand, you get isotropic expansion of all the genomic DNA fragments that are there. So all the dumbbells are now in their own position. They don't change the position. And you get a isotropic expansion. And next step we do is do a rolling circle amplification. So in the end, what you see is we call these colonies, but these are colonies of DNA, unique DNA fragments containing the genomic DNA and its associated barcode. And there is the key. Now we can sequence each of these colonies using Illumina sequencing chemistry on the microscope, right? So there is a barcode with each of these ROLNI. And when we do the Illumina sequencing chemistry, we get a fluorescence and we record this fluorescence in multiple rounds so that we can finish reading the whole barcode from each ROLNI. Usually, typically our barcode is 20 to 22 base pair, and that is enough for us to decode each and every cell and all the ROLNIs after the sequencing is done. So then this is how the sequencing works. This is all done on microscope. Every round we sequence one base, and this way we can sequence multiple cells on microscope. So you see the color and the ROLNIs. So we are now decoding the entire barcode. After the sequencing is done, what we do is we take these in situ sequence cells. You can see here the barcode. This is unique molecule. Here there is a 21 base pair. And then we can decode each unique molecular identifier and have a barcode decoded. But what we don't have is a genomic DNA sequence associated with that barcode. So for that, what we do is we take the whole DNA out of the nucleus, out of all the cells, and then we ex situ sequences. So for ex situ sequencing, we have a PCR primer that can allow us to extract the genomic DNA along with the barcode associated with. And then when we sequence outside, what we get is from the in situ sequencing, we have a in situ barcode and we have its position, XYZ position. From the ex situ sequencing, we have genomic DNA. And now we can match these together by our mapping algorithm so that we now have XYZ position of each and every DNA fragment. And I should tell you that each DNA fragment in each of these ROLNIs is around 100 base pair because our tagmentation frequency is roughly around 50 to 100 base pairs. So each piece of DNA here is just 100 base pair, which you typically don't see in FISH techniques, which is usually in hundreds of kbs in FISH techniques. Right. So now our task is to take all of these ROLNIs that you see here. They are colored according to chromosome in this. So we have a chromosome clustering algorithm where we can filter out the reads that were not sequenced properly or the reads that or the fragments that have moved really away from the chromosome. So there is an algorithm that we use, which is chromosome clustering, and that allows us to form chromosome structures. And this is how it looks after the sequencing and mapping is done. So you can clearly see the chromosome architecture and territories. Next, what our challenge is to now associate each of those dots that we saw with its associated mark, whatever we want to label. So in this case, we have labeled lamin AC K9ME3, H3K9ME3, which is a repressive mark, and SC35, which is an active mark, which is a speckle. And on the right, you're seeing a pre and post expansion. This is how we calculate the expansion factor, because we also have to find out the real distances in the cell from these proteins. So we have to have an expansion factor per cell. So we can calculate expansion factor per cell because we have pre and post expansion images of the same cell. And what we can now do is we can trace the chromatin, the DNA fragment with its protein. So here, what we are seeing is the distance to lamin in this case, or distance to immunostain. The dark green means it's very close to the immunostain, and the faint green means it is away from the. And now this is basically acting as a chip, that is chromatin immunoprecipitation in situ, where we are seeing the DNA fragment associated with its protein. And what we can do is we can then find out the distances for every bin. So here in this case, we have used 500 kb bin. So we can bin the chromosome into 500 kb bins and look at its distance from the particular mark. And what we found is that it's very well correlates with the attack score, which means attack means more open chromatin. So we have attack for the same cells, and it tells us the attack score of each of the bin. more the attack score, more open that DNA fragment is. And what we can see is that in case of lamine, it's separating from the lamine contact frequencies, whereas in speckle, which is active, the attack score is very well correlated with the distances. That means it's working. The distance correlation is working. So now we can literally trace the chromatin that is associated with each mark in situ, and not just trace, but also look at its 3D position. Okay. But now, can we use this for real life biology? This is just the example that I showed in IMR90 cells that we did all the validation, but can we use it? So we decided to use a very well-known model, which is progeria, in which we know that there's a mutation in a laminin protein, and that mutation causes nuclear disruptions, the nuclear deformities, and that also leads to a very drastic phenotype where you can see accelerated aging in these individuals. They usually don't live longer than 20 years. We also have another model, which I'm not going to show today, which is partial reprogramming, where we wanted to change old cells to young cells by expressing OSKM in them, and then see if we can detect changes, nanoscale changes in genome organization when we express these OSKM markers in these cells. But I'm not going to talk about this. Progeria is very simple, and you can literally look at these deformities at a very small scale. Okay, so for that we took normal fibroblast cells. This is the mom of a progeroid, normal mom of a progeroid child, and these are the different passages of the same progeria cells. So as you know that when we passage these cells, they age very fast, and up to passage 26 or 27 they die. So this is a very late passage. This is a very early passage. And between these passages, they have aged a lot. So we wanted to see if we can detect this difference during when they age. So we did an IGS, in situ genome sequencing, on these cells, normal progeria, normal pyroblast cells, early progeria and late progeria. All these dots are sequenced genomic DNA fragments. We have multiple cells for these. And this is how their genome looks. So first thing that we wanted to do is to see whether there are kind of large scale changes in genome organization. So what we did was we looked at the chromosome architecture in these cells, whether it is disturbed. So this is a control fibroblast cells, and you can see here the chromosome is kind of intact. It has its own territory. On the bottom, you're seeing a 3D look of the chromosome, and the line here depicts the cross section. And you can see these two chromosomes, chromosome 10 and chromosome X, are nicely situated at the periphery in their own zone. Whereas when you look at the a progeria cells from passage 22, first thing that you can notice is there is a lamine invaginations. These lamine protrusions literally can go from the one end of the nucleus to the other end, and they form these tunnels. We call these lamine tunnels, lamine nanotubes. What they do is they disrupt the chromosome organization. You can see it has gone through the chromosome X, and it's not allowing it to form its own territory. In other cases, what it can also do is it can actually capture the entire chromosome in a small section, small region. So it is causing a global disruption in how the chromosome is organized. So we can now detect these small scale changes, or these large scale changes. Not only detect, but we can sequence a specific chromosome associated with it. But the next thing is, we wanted to know if this change, let's say, for example, in this cell, there is a change in chromosome 8 organization or chromosome X organization, but what happens to the other chromosomes? Are they all disrupted? Because there is a relatively normal part you can see in this cell, even though this is a progeria cell. But except this invagination, there are also chromosomes located in these normal parts. So we wanted to know if this change that happened due to these nanotubes is it local or is it global? And our technique can allow us to do that. No other technique so far can allow to look at the local or global changes. So we wanted to see if those changes are local or global. So for that, what we decided to do is to segment the lamine into internal lamine and peripheral lamine. What I mean by that is when we see the invagination, We can now segment the lamin internal, which is located in the nucleus, going inside the nucleus, or we can also look at the peripheral, only peripheral lamin. And now on the right-hand side, what you're seeing is we are trying to associate the read with internal as well as external lamin. So now you can separate the chromatin that is with the internal lamin or with the external lamin. So now what we can do is, like I said, we can segment here you can see the segmentation. All the pink is internal lamin. In the control cell, there is almost 0% internal lamin. But as the cells age, they show more internal lamin. And now here in this, you can see in this graph that control cells are all clustered here. They have very less internal lamin, whereas as they age, they accumulate more and more internal lamin. So we can segment them very well at nanoscale. It's a very small, very, very small scale. And this is the segmentation and association of the Rolneys with the lamin. This is a normal cell. You can see top, middle, bottom, and each of these Rolneys, the pink, the green depicts the distance to the lamin, external lamin, and pink is internal lamin. And that is completely disturbed, as you can see in this progeria cell, where you see more and more chromatin getting associated with internal lamin. Now, what we can do is now we know the chromatin that is with the internal lamin. We know the chromatin that is with the external lamin. Can we characterize it? Can we find out what type of chromatin that is? So what we have done in this case is, let's say, for example, here, what we are seeing is a graph where we plotted we converted chromosome 5 in this example into small bins. These are 500 kb bins. And for each bin, we look at its distance from the lamin. In the blue, you are seeing a distance from the peripheral lamin. That is the contact frequency. Okay. And we also plotted GC content of that particular bin. Why GC content? Because GC content depicts the activity. More the GC content, more the active chromatin is. So what you can see here, for example, in this region, you can see higher GC content, more active chromatin, and less contact frequency with lamin. That means this chromatin is not contacting the lamin. That is a peripheral lamin. But now, when we go and look at the internal lamin in the same cell for chromosome 5, what you can see is that this part of the chromosome is now touching internal lamin. Although it was active, it is still touching the lamin that is abnormally located. So we did this for every cell, and not just the GC content, but we found the attack score. We did a attack in normal cells, so that for each fragment we can have an attack score from normal cells. So we compared the attack score of each of these small bins, 500 kb bins, for all chromosomes and all cells. On the y-axis here are all the cells. You can see horizontally, these lines are basically all cells. You're looking at all the chromosomes, 500 kb bins. What blue to yellow depicts is the distance to lamin of that each bin in each cell. And we arrange these cells according to the percentage of total lamin they had. So the cells at the bottom had more lamin, cells at the top had less lamin, total amount of lamin. Then we did an attack score analysis where each fragment was scored with its attack score, which is compared with the normal from where we took the attack score. And then we compared that attack score with the lamin contact frequency, and we call it a disruption score. So when the chromatin fragment that is not supposed to touch the lamin, if it is touching the lamin, it has more lamin attack correlation. It has less lamin attack correlation score. Okay, that means that that chromatin is more disrupted. It was not supposed to be with the lamin. And that way, when we cluster cells according to disruption score, we found that all the cells that were from like progeria passages, they clustered at the bottom, whereas the control cells clustered at the top. That means our technique is very well working, even blindly clustering cells based on their attack score and lamin attack correlation score, which is a disruption score. So, so this was done for all the chromatin fragments everywhere in the nucleus for all the cells. But we wanted to know now the nucleus is huge. And we wanted to know if there are good neighborhoods and bad neighborhoods in the nucleus. So we wanted to know if it is local or global disruption. So for example, in this Progeria passage 22 cell, there are two neighborhoods, neighborhood 1 and 2. What we can do is we can look at these two different neighborhoods and find out attack score for each of these chromatin fragments. So on the y-axis here, you can see attack score from the normal cells that we are comparing with and distance to lamin. So each of these dots, we can compare their distance to lamin. So more they are away from the lamin, higher the attack score. That means they have more open chromatin, and that is from the norm for a normal cell, right? So in this neighborhood, this which looks very normal to us, you can see that there are more fragments that are away from the lamin and they are more open. That means they have more attack score. Whereas in this disrupted neighborhood, you can see that more and more fragments are located zero distance to lamin. That means they're very close to the lamin. They're not supposed to close to the lamin, but they are. That means there is a local disruption of the chromatin, and that is why what we call as a hotspot, where there is these lamin invagination or a small scale lamin tubes, nanotubes, which which we usually don't see with normal microscope, but with expansion microscope we do, and we can now assign chromatin fragments to those and find out these hotspots. So we call this as a hotspot analysis. So we do see, we compare it with control, and you can see that in control cells, there is a more lamin attack correlation. That means there is more organized chromatin, whereas in progeria cells, you do start seeing that the lamin attack correlation is very low. That means there are more and more disruptions due to these internal invaginations of lamin. Here also, when we arrange cells based on these local hotspots, you again see that the cells are very well separated or clustered, normal cells on the top and progeria cells on the bottom, and the attack score is shifting towards left. That means it's becoming more and more disrupted. And same is in the right hand side graph, where you can see the control cells are nicely clustered, having more attack score, whereas the lamin or the progeria cells are all over the place. That means they have more and more local hotspot or disruption. In our analysis, we also found that this trend that we see of local disruption is not uniform. Every cell has a different kind of chromatin that is disrupted. That means it is very random. Whenever there is a accumulation of lamin inside the nucleus, we see that it is completely random. It is just touching any part of the chromatin and causing chaos in the nucleus. And that is why these cells are extremely chaotic, very hard to just do a fish imaging or something to look at a specific genomic region. There is no specific genomic region. It's extremely random. Now we wanted to see what is it doing by contacting this chromatin, which is not supposed to be with the lamin. So as you can see, we also notice that there are sometimes, occasionally, normal cells also show these lamin nanotubes. And you may have seen, whoever has done the lamin staining, you do see these internal lamin foci. When we did a polymerase 2 for RNA polymerase 2 staining, this is an active polymerase 2, serine 5 phosphorylated, mostly associated with promoters. We saw that wherever there is a lamin dots inside the nucleus, it has an exclusion zone for RNA pol 2. There is no RNA pol 2. So we hypothesized that maybe in progeria cells, these lamin nanotubes protrusions are causing non-specific repression of active chromatin. So that is what we did in here, where we stained the lamin and did expansion microscopy for RNA pol 2, and we found in normal cells, as you can see at the periphery, there is a heterochromatin, there is a RNA pol 2 exclusion zone, means lamin is repressing the chromatin that is associated with it. Whereas in progeria cells, it's also repressing the chromatin that is internal, which is supposed to be active. So we call these as a non-specific repressive zones caused by these accumulation, a small accumulation, even a small nanotube of lamin. And we found evidence that these protrusions or invaginations of lamin are also true in aging cells. So we did this analysis in aged cells. So this is all done. So we took a mouse heart tissue from aged mice, and we found that these invaginations are causing a lot of repression of active chromatin in aged cells. So it is all non-specific. Every cell has a different repression. We also took a 92-year fibroblast cells. These are normal cells, not progeria, and we do see that older fibroblasts have these invagination causing exclusion zones of RNA pol 2. That means that part of the chromatin is repressed non-specifically. With this, we have come up with a model with very high resolution technique that allow us to look at extremely small scale changes in the chromatin. Currently, we focused in this paper on lamin, but in future, we can in fact look at very small other nuclear architecture proteins, or we can also look at other disruptions in the domains of euchromatin and heterochromatin and look at a specific DNA that is inhibited or disturbed, or we can look at the transcriptional activity of that particular chromatin with this technique. And with this, I would like to conclude. So this is my lab, and these are the people who are mainly involved, who is Zach, who did all the computational analysis, and Caroline is working on experimental part with me. And this is Jason, who is our supervisor. With this, thank you so much, and feel free to ask questions, or if you want to write me afterwards, that's fine. I'll be very happy to answer every small details of this protocol. Thank you.
Thanks, Ajay. That was fantastic.
Thank you.
I open the floor now. If people want to ask questions, they kind of mute themselves or put them in the chat. While people are thinking, I was wondering about the expansion factor. Does the addition of the sequencing changes how much expansion you get, or is still around?
Yes, that is a very good question, and I forgot to mention that after we expand the sample, there is a process called as re-embedding. So we take the expanded gel and re-embed it into a secondary gel, which is only made out of just water and acrylamide and APS and TMID. So it doesn't have any salt. So our primary gel, which was expanded, doesn't get shrink. And when we re-embed this, we use a siliconized accredited glass. So we usually use bind silane, which accredites the glass. So you treat the glass with bind silane so that when we polymerize the second gel, it's fixed to the glass. So now it is completely fixed and never changes its expansion factor. It just fixed. And we have to also do a process called as passivation, gel passivation, so that all the enzymatic works because the gel has a charge on it. And when you put the enzyme active sites are also charged, so they are inhibited. We have to passivate the gel by removing the charge. So there is a process of passivation, and we have it in the paper. But yeah, I'm happy to answer any kind of questions regarding that.
Let me think. And the other thing I was thinking, is there any modifications that might be responsible, like maybe methylation or something that could be responsible for things that are...
That is, yeah, we did try that. We wanted to look at DNA methylation in situ. The problem with all the DNA methylation techniques currently is that they are very harsh. The treatments are very harsh to convert the DNA methylation. So we did try that. It didn't work. It actually distorted the DNA morphology. the nuclear morphology. So we didn't use that. Yeah. But there are other enzymatic ways which we have not looked at.
It's very cool.
Yeah.
Yeah. Any questions from the public? Oh, here there is one. You want me to read it out, or are you happy to read it?
Yeah, it's for everyone. Yeah.
Hi, Ajay. The work is awesome. How can one make sure the digestion doesn't alter the lamin structures or orientation after expansion? Any comparative analysis possible?
Yeah, very good question. We have tried different digestion conditions, and if you look at the expansion protocols, there are various kinds of digestion people have used. For us, we have fixed on a one set of protocol which doesn't alter the lamin or any other stain, and you can find it in the protocol. But we have optimized it. I have tried different dilutions of proteinase K, and even if you change a small part of it, if you, let's say, for example, we do overnight proteinase K digestion at, I think, 8 unit per mil of the proteinase K. And if you change it to 37 degrees Celsius, you will see that everything is completely gone because the proteinase K is extremely active. So you have to be very careful when you use the proteinase K in this case. We do not have comparative analysis from our data, but you will definitely find papers. I think there's one paper on C. elegans where they use actually different enzymes, not just proteinase K, to compare how the digestion works. But yeah, we do not have ourselves as a comparative data.
What type of software were you using to align, I guess?
It's all done in MATLAB by Zach. It's the code is there in the paper. I am not a computational expert, but I know how it is done. I don't
Yeah.
have the code itself. But it's pretty much using the code where you just take pre-expansion images and try to use matching algorithms to match pre-expanded shapes to post-expanded shapes and spatial positions. So you have relative distances between different cells. So even if you find one cell, you can find all the cells very easily. Yeah.
Yeah. Any other questions? Otherwise, I can ask forever. Sorry. The other thing you were mentioning is about this process, how it's random, like the chromosomes would
Yeah.
be in different locations every time.
So what I mean by random is that ideally people expect that a certain chromosome or a chromatin fragment might get associated with lamin,
Yeah.
and you just do a fish for that in all cells. But that is not true in all cells in case of in lamin invaginations. These lamin invaginations are physical. They do not have specificity towards chromatin. They just randomly form because this is a disease and they just keep on accumulating randomly. So it's very hard to pinpoint a specific fragment of chromatin because we do see that it's everywhere and it's very random in each cell. So it is very heterogeneous in terms of its contacts. Yeah.
We'll leave it there. If anyone has questions, feel free to email Ajay, or you can also email me. I can give you his email. Thank you very much for joining us.
Thank you. Yeah, it was nice
Right?
talking to all of you and very welcome. You can always email me for anything. Yeah, thanks.
Thank you very much.
Right. Have a good one. Bye.
Bye.